All right, the purpose of this video is to walk you through Activity 10-3, Understanding Protein Electrophoresis as a Diagnostic Tool. I give you the gene sequence, just in case you're interested, the way it looks in the NCBI database. And so the HBB gene starts with an AUG here. The gene sequence is written in DNA though, so a TG would be the start codon, but you remember that the gene is more than just the protein coding sequence. And then down here is the stop codon, TAA, which in RNA would be UAA. And so this translates to a protein given here by the single letter sequence. And I give you the chart here that shows you how the single letter abbreviations correspond to the three-letter abbreviations and the full names of the amino acids. So if you look through this protein sequence for hemoglobin beta subunit, you'll find that there's 23 basic amino acids. The basic amino acids are lysine, arginine, and histidine. So if you look through the protein sequence, lysine has a symbol of K, arginine has a symbol of R, histidine has an H. So if you went through and counted up those letters, total of 23. The acidic amino acids are aspartic acid, which has a symbol of D, and glutamic acid has a symbol of E. So those are kind of the important things that we're looking at, because we're looking at the charge of the protein at different pH environments, because that's the trick that we're using when we run the blood in an electrophoresis gel. And when you have a drop of blood, you have tons and tons of hemoglobin, because red blood cells are the most abundant type of cells in the blood sample, and red blood cells are just loaded with hemoglobin. So you have plenty to load onto a gel. It's a very convenient way of doing this kind of diagnostic test. You don't need much blood, you don't need much materials or equipment, and you can do this diagnostic test. But in any case, so I need to teach you just enough protein biochemistry for you to be able to answer some of these questions. And you may recall that a protein has, I'm going to see if I can get a whiteboard here. Let me open up maybe a PowerPoint. I'm going to show you a little something here. All right, so let's say let's say you have an amino acid. You have for any amino acid, you have the amino group and you have the carboxyl group. And there's a variable chain, which we usually call R. So this is the amino group, and then the carboxyl group is over here, C double bond O, OH. So this is the carboxyl group. So when you have a protein, you have a whole chain of these, but I'm just going to show you what happens on one as an example. If you put the protein into an environment that has a very high concentration of hydrogen ions, which means if it has a very high concentration of hydrogen ions, that means it has a very low pH. So that's the definition of acid. And if you do that, then what's going to happen is the extra hydrogen ions, the protein itself will try to buffer the solution. It'll try to counteract the environment. And so it will try to absorb some of the extra hydrogen ions. So it will pick up an extra hydrogen here. The carboxyl group won't do anything. So overall, your amino groups will pick up extra hydrogen ions, and overall the protein will take on more of a positive charge. And that's if you're in an extremely acidic environment. So not only will the end amino group take on a hydrogen but an extra hydrogen, but any of the R groups that have an amino group. So amino groups are found in basic side chains. So what that means is if you're in a very high hydrogen ion environment, a very acidic environment, then, so in an acidic environment, the acidic side chains won't do anything. It's the basic side chains that will be ionized. Basic side chains will be positively ionized. And acidic side chains, carboxyl groups, will stay neutral. The opposite will be true, though, in a basic pH. So let me draw the same amino acid, NCC. This represents kind of our protein. In a basic pH, there's a very low amount of hydrogen ions, and that means a very high pH. So we're using pH 9.2 in this lab. And at a very low hydrogen ion concentration or a very high pH, again, the protein is going to try to buffer the environment. And the way it'll do that is to try to raise up the hydrogen ion. So what it can do is release this hydrogen here. That can be released into the solution to try to neutralize the pH. Nothing will happen with the amino group. So in a basic situation, in a basic buffer environment, it's actually the acidic side chains will ionize in a negative way, acidic groups. And not only the terminal carboxyl group, but also anything on the R group will ionize negatively. So in an acidic environment, the protein will become more positively charged. In a basic environment, the protein will become more negatively charged. And in a basic environment, the amino groups don't ionize at all. It's the carboxyl groups that ionize negatively. So that's just enough of the protein biochemistry to help you to answer these questions that I've set out for you on this section of the lab.